extremely quickly, let's go through and recap, okay? Antibodies, we have 5 different isotypes. The main way I like to remember it is gamed. IgG, IgA, IgM, IgE, IgD, right? They have their corresponding Greek numerals here, right? So we have this gamma, we have alpha, we have mu, we have epsilon, and we have delta, okay? Let's come over here. Over here we have a secreted antibody and a membrane-bound antibody, and we're going to talk about their differences. But quick anatomy real quick. A secreted antibody has what? It has this heavy chain, right? This heavy chain, also a heavy chain, and a light chain on the outside, okay? Now, these heavy chains and light chains, they all have their own specific region. They have a constant and a variable region. Here is a constant region of the heavy chain, constant region in this black shaded in portion. This purple portion, this is our variable heavy chain. Okay, variable heavy chain here, variable heavy chain here. Now the light chain, it has a constant light chain and a variable light chain in pink here, okay? Now, another thing you have to know is that the combination of the constant heavy chain, constant light chain, variable heavy chain, and variable light chain is known as the Fab region, right here, Fab region, and it is also the antigen binding domain. The antigen binding site, okay, is what, guys? Is the variable heavy chain and the variable light chain. And it is made up of, it has these complementarity determining regions, also known as your hypervariable regions, okay? The constant heavy chain down here is known as your FC region. FC region can be used for complement fixation and phagocyte binding, okay? So now that completes the anatomy that we talked about. Again, we also have this hinge region here, and these green things here in between are these disulfide bonds that hold this structure together. Now, we talked about the complementarity determining regions. So let's come back here. The complementarity determining region is where the sequence variation occurs. So this is really where that specificity of the antibody is generated. Okay? And so that region is also where the antigen is going to interact with the antibody. The FC region of the constant heavy chain, okay, remember, this was the FC region of the constant heavy chain. This is going to determine the differences in your antibody isotypes, right? Because we have 5 isotypes. So this is going to determine that difference and their effector functions, okay? So this is why each immunoglobulin has a different effector function. Another important thing, you know how I said we have a light chain here and a heavy chain here? The heavy chain has 1 isotype. That's it. The light chain has 2 isotypes. It has a kappa and a lambda, and they have no effect on the immunoglobulin isotype, whereas the FC region has an effect on each of these guys' isotypes. The kappa and the lambda, they have no effect on the immunoglobulin isotype. And just remember that each B cell can only have 1 isotype at a time, 1 light chain isotype at a time. Okay, so I can't have a kappa and a lambda here. It has to be a kappa, kappa. This one, for example, this could be a lambda and a lambda. Okay? Okay, that gets that done. So now let's quickly go and recap the membrane-bound version of the immunoglobulin. And really the only big difference is that you have a longer FC region because it has to reach the plasma membrane. And the other thing you have to know is that it has these invariant membrane-bound proteins called immunoglobulin alpha and immunoglobulin beta, and they're used for signal transduction. They basically recognize when this membrane-bound immunoglobulin has bound its antigen. Okay, they basically recognize that and they say, oh, okay, it's time to send the signal to the cell. Okay. All right. So now that we understand most of this thing, let's very quickly just go through the different types of immunoglobulins. We have an immunoglobulin A, which can have A1 and A2. It is mainly in the dimer form, right? Dimer form, but it can be a trimer and it can be a monomer. And again, those dimer and trimers, they're held together by this J chain, okay, J protein. IgA is responsible for mucosal immunity. IgD is a membrane-bound antigen receptor, right? So it's a B cell receptor, and it's mainly in the monomer form. Immunoglobulin A gives us helminthic protection from these parasites, and it gives you an immediate hypersensitivity as well. Again, also in the monomer form. IgG, one of the most important antibodies in the body. If you have to remember one, just remember this. It has 4 subtypes: IgG1, IgG2, IgG3, IgG4. Okay, and it can be used in opsonization, complement activation, antibody-dependent cell-mediated cytotoxic function. It can be used in neonatal immunity, meaning that your parent will actually give you an IgG that passes through placenta. Okay, and it can be used in feedback inhibition of B cells. IgM, the IgM antibody, in its secreted form, it takes a pentamer shape. Okay, in its monomer form, it's membrane bound, and it is a membrane antigen receptor. Okay, so it's a B cell receptor. But when it's secreted, it can do complement activation. Okay, and it's really, really good at complement activation. Okay, it's in fact one of the best ones. Another thing I want you to know. Okay, just the last couple of things. Last things I want you to know, and we're done. You can see this red thing here is the antigen, and this thing here, this blue thing here is your antibody. The site where the antigen binds to the antibody, okay, is known as your epitope. Okay, it's known as your epitope, and they have to complement the antibody. If it doesn't complement the antibody, this is not going to work. Right? So I have an example here. This is a triangular antigen trying to bind to a rectangular antibody. This is not complementary. You cannot bind. Another thing you have to know about epitopes. Epitopes can be discontinuous, okay, meaning that this protein here is not in a linearly lined fashion. It's discontinuous, right? I am binding antigen from this side and another side. A linear epitope is when I'm binding it from the same chain here, okay? So just understand that. And each epitope can have a different sort of conformation, which actually leads us here. If an antigen is denatured, okay, an antibody might not recognize the conformational epitopes on a denatured antigen because it was originally generated for the native form of that denatured antigen. Okay, so just understand that it might not recognize it. And again, what does denatured mean? Denatured just means that it's been, acidified or whatever, and it has changed its 3D conformational form. Okay. All right. Last thing I want you to know is the affinity maturation. And the affinity maturation basically is when the immunoglobulin's affinity to bind to an antigen increases because of a second exposure to that antigen or that pathogen. Okay? And that basically completes everything.